me try. Okay, now let me share my screen. Oh, I hope everyone can see it now in the presentation mode. Yeah, please let me know if not. Yeah, again, as Melissa said already, I'm Dieter Hennecke. I'm the coordinator of this residue project. And if you see me watching to the side from time to time, the reason is because there's my screen and there's my camera. So if I want to see my own slides, I have to look from time to time to the side. I would like to give you now a brief overview on the project, on the objectives of the project, and also on some results so far. But this will not go into depth, because for details we have later some talks from the other partners which are connected to the project. They will give you more details about project results so far and what is about to come. First of all, I want to tell you, or want to give you some idea about the framework where the project is located. We are in the PRIMA project, or the project is running in the PRIMA framework, and all this is under Horizon 2020 in the EU program. And I highlight here in blue, I mean, this is about PRIMA, what PRIMA says about them itself. Sorry, what was that? Anything for me?
No.
Okay. I highlight here in blue what is most important for our project. It is a science program, and we develop an innovative solution for water provision and management in the Mediterranean area. That describes our project pretty well already. And in PRIMA, you have 19 countries, not only EU member states, which is really good, because it's really good to work with other countries together in the Horizon 2020. Must not be always EU countries. Let me see. Yeah. So the problem definition, I will make it very brief only, because I think Dr. Mauer will later say a little bit more about that. This is only the process. I mean, what you can see on the figure is the process how anthropogenic organic contaminants can reach the agriculture, the agro environment, agriculture. So that means it goes down the drain and via sewage sludge and irrigation water, which you have to use sometimes for wastewater you have to use for irrigation, because you have no choice. There is not too much water. That is the entire problem of all this. The contaminants can reach the soil, and via the soil they can reach, or they can be uptaken by the crops, and via the crops it comes back. You see this the arrow here. It comes back to you or to the urban environment, and then you have risk of food contamination. And all this is not theoretically, but this is already proven by several studies that this really happens. Yeah. And our project goal is to improve the safety of these agricultural products, which are grown in countries which might have this problem, because they have to use wastewater for irrigation, because there is not really a choice. What we want to do in our project is not setting new limits, because that is always a problem. New limits always brings new problems. But we want to develop a technology which reduces simply the contamination in soil, and which improves also the agricultural products, but also the soil. If we improve the soil, we might be able to improve also the agricultural products coming from this soil. And the way to do this is a production of new production methods for safe soil amendments, and these amendments should also help to detoxificate the soil, to reduce the contaminant load that we cannot avoid, or that we might not avoid by using the water for irrigation. The project partners, I saw already others have the slide too, so I'll make it also short. Is two partners from Germany, which is us, Fraunhofer, and the University of Berlin. We have a partner two partners from Israel, one from Italy, and one from Spain. Yeah, I think that's it. And each of them will give a later presentation about the part what they do in the project. So I don't want to do more about that. To the project structure, I want to go a bit more into depth because this shows also what we wanted to do. This explains a bit the project and explains also where we are and what is still missing. And as you can see in work package 1, the work package 1 was designed to bring all the source materials. Yeah, we needed the source materials. What do we want to do? We want to use local waste streams. So we had to establish, okay, what kind of waste streams are available in the areas of concern, or in these areas of the countries where we want to apply this method. So first, we needed to check, okay, where's wastewater treatment plants? What happens to the sewage sludge? And where can we get this sludge, which we want to use later for this for making the substrate? We had to characterize this sludge and to see, okay, which are really the relevant contaminants where we can look, because later we want to take also some examples, some reference substances to see if this method really works on the relevant contaminants. So we had to select reference substances. We had to purchase reference substances. You will see that later a bit more into detail. I go into this a bit more to detail. We had to get sewage sludge, and even more important, we had to ship sewage sludge, and it's not so easy to ship sewage sludge from Israel, for example, to the European Union. That's already the first challenge. In work package 2, we took this, or I can say, because it's done already, we took this waste materials and tried to make something new out of it, which is a biochar. This will be presented later from Dr. Drapkin and René Schatten. And with this biochar, we can amend it with sewage sludge and produce a substrate, which in the end, this should be more or less the magic substrate, I should say. This is green, I don't know if I can show it. Ah, yeah, there's a mouse. Here you see, this is really the substrate we want to develop, and then later we will test if the effect is what we expect. One part is a viability testing done by the Spanish partner. We do a greenhouse trial and a bit bigger greenhouse trial in technical scale. Since there's an outdoor lysimeter study in Israel, directly there in the location where we want to apply it later, so a bit bigger, up to a field trial, and all this runs in parallel here, and we will see if the effect will be as expected. So we have different scales of trials, and they will be all very related to practical application. Of course, in the end, you have an evaluation, risk assessment, and so far more important is now the communication, which is not only required by Horizon 2020, but also it makes really sense to communicate your science. Science that you don't communicate doesn't happen. So it's very important to have, for example, we have a project website. We are in social networks, in Twitter, I think we have an account. We plan open access publications. So far, we do not have publications, but it is about to come. And knowledge transfer, also that very important. Yeah, we don't want to run like so much research to end up in the in the valley of death. Yeah, it's so called because you have we make a great research and it could be applied super good, but it never reaches the persons who apply it later in reality. Yeah, that is the valley of death is called like that, and it happens really to very much research that in the end no one will ever use it. And to avoid that, we are already connected with the players who apply it already, and we also plan later then on-site field days and position paper to inform also the politicians. This is also important about our research, that they can probably do the next measures to support it, because it should be good for everyone, and so everyone should be informed about that. This is the current project status where we are. Everything in yellow is what we more or less did already. Yeah, you can see that, and which is not entire yellow. This is where we work on, and the reason is why that we have not really the substrate ready, or probably is ready now, but it's not distributed between other partners. And that is why bioavailability, Dr. Ortega Calvo will present that later. We did a lot of work, but not with the substrate because it's only available right now. And this is also why we could not start the greenhouse trial or this trial so far, but it is directly in front and we will start very soon. What we did already is, as you can see here, this arrow, this yellow arrow, we shipped already local soil. Also, this is really a challenge, yeah, because we don't talk about 20 kg or so, but we talk about 9 tons, what we shipped from Israel to Germany to make a greenhouse trial in technical scale. Also from the communication, you see already the summer school, that is what you face now already. There will be another one in fall. And also the participation of national and international events. We are already in workshops, in international workshops, presenting our study. And yeah, we will see. I mean, it's always better when you have results to present it, but also at this stage to see where we are and what we do. Sometimes you get nice additional input. When we go a bit into depth, the project status, work package 1. No worries, Dimitri, I will make it very short here. This is only about the composite sewage sludge, which we use, which we shipped already from Israel to FU Berlin. And this sludge, it was not so difficult to ship because we could autoclave it before, we could sterilize it. And sterilized material you can transfer over borders, yeah, which is totally different with biological material. That is really difficult to transport from one country to the other, in particular outside of the EU into the EU. Yeah, so here we didn't have a problem because this sludge was, or this compost was anyway meant to be carbonized. That means for biochar production, and it is also. No, I go one front first. This is also what we did here. The carbonization. I think Dimitri will show that later in the more in the talk. We did it here in the process a bit. Yeah, I think I don't need to explain it more detail. The challenge here was really later when this biochar should be amended with fresh sewage sludge. That is not possible. Yeah, it's not so easy, or not even so easy, but it's almost impossible to ship fresh sewage sludge from Israel, for example, to Europe to make their experiments with composting. And also we saw later the compost process was affected by the biochar. But okay, this is something for later for another talk. Let me go back to this work package 1, what we finished. This is a reference substances that we selected after checking the biochar, the sewage sludge, after checking the literature which is available, and it's carbamazepine, which is an anti-epileptic, or the anti-epileptic, I must say, is widely used, and you find it really in relevant concentrations in the wastewater and also in the sewage sludge later. And is lamotrigine, which is an anticonvulsant. That means it helps when you have cramps, for example. Yeah, for everyone who is not a medical doctor. And also this is really, yeah, it goes normally down the drain. Yeah, normally when you take drugs, it is a huge part of it is also excreted, and then it ends up in the sewage treatment plant, or in the sewage sludge, or in the irrigation water. And this is why they are important. They are labeled, radio labeled, as you can see. We radio label them in the most stable part of the molecule, which is because when you see this, the carbamazepine, you can also label it here in the carbonyl position. But the problem is that this carbonyl position could be split, and then another part which is supposed to be stable or could be stable becomes invisible. That is why we label only in the most stable part. And if this little part breaks here, there's no concern because this will degrade pretty fast in the environment. But this maybe not. Yeah, this part might be stable and might be also taken up. And that is also the reason why we use a radio label. First of all, we can make a total mass balance. We simply look how much of the radioactivity is later still present and where the rest is gone. And on the other hand, when there happens some transformation, some biological transformation, for example, or an uptake to the plant or so, you cannot trust that it's the parent molecule anymore, but probably only part of it. And that is why I say this most stable part, because when it will be degraded, it is assumed that this part will stay the longest. as it is. Yeah, and then we can look simply for the radioactivity, and we can follow this by this radio labeling. We can follow unknown pathways, because if you know, probably when you are familiar with chemical analytics, you will only find what you are looking for. And if you don't know to which products it will degrade, you will never find an unknown transformation product. With a radio label, you will find it. You simply follow the path of the radioactivity, and then you can look with sophisticated chemical analysis like high resolution LCMS, GCMS, NMR. Everything is possible. Then you can look, okay, what is it? What do we have here? What kind of chemical is it? Is there any concern or is there no concern? Is it simply a biological molecule? Because when it's degraded, sometimes it's also built into the biomass. Yeah, this all we can look, and that yeah, justifies the use of a radio label. I can tell you, these radio label substances are super expensive. Yeah, we purchase them both from a provider, and we're talking here about a few milligrams each. Each of these molecules is a few milligrams. Let's say, I think 100, 200 milligrams, and they cost more than 60,000 euro. So it's really very expensive components. But the only way, really, the only way to follow a substance in a matrix, yeah, in a complex matrix. And that is why this 14C is, for example, also mandatory in pesticide regulation. And that is where we come from. Okay, this we had already. Work package 3, the bioavailability. I don't want to tell more because José Julio will do that later. Work package 4, that was already the greenhouse trial. I mean, we have already sampled and import the 8 tons or 8 or 9 tons, I'm not 100% sure. And we started ready to develop the analytical methods. I have some photos from the soil sampling. The challenges were here a bit unexpected because it's not always peace in that area. And I remember that we had to delay that for a few weeks because they were fighting at that moment in this area, and we didn't want that someone gets hurt. Fortunately, they stopped fighting very fast or very soon, and then the sampling could happen. And as you can see here, this is not a little sample. This is a big sample. The shipment is all in Israel here, and then it arrived in Germany. You can see our greenhouse, and in the end, here on the right side, you can see the plots which are prepared is always half a square meter here, and we have individual plots where we can apply different crops and where we also can apply the different reference substances. Sometimes radio labeled, sometimes probably not, sometimes probably with a control. Yeah, I think we have it in the end, we have more than 8 different plots. So of course, we could start already with the development of the analytical methods. We have radio HPLC, we have radio TLC. That is the normal analytics you do. Just for showing you how this works with the radio HPLC, here's a UV peak, and here's the same chromatogram with radioactivity. And as you can see, all these little matrix effects here. I mean, even if it was simply from solvent, but you can see something here. In radioactivity, you don't see that. In radioactivity, you see only the radioactivity. And if you have here a matrix loaded sample in UV, which have hundreds of signals, in 14C detection, you will not see any other signal. You will see only the one from the parent, only from the radioactivity. Or if you have one degradation product, there's probably, or hopefully then, one other signal. Yeah, that is the purpose and the advantage of the 14C detection. So what we did already is trying to get the radioactivity out of the plants. You can see here the alfalfa plants. The way to do it is you put simply, so you take the plants and put it simply in a solution with your radioactive substance, and by evaporation, they take the water up, and then you have the radioactivity in the plants. That's the easiest way to get it in the plants. And later you make you look, okay, what is back here, or what is left over in the vessel, and then you know exactly what is in your plants, how much radioactivity you have in your plants, and then you can try to extract how much of this radioactivity you can get off. Yeah, and that is a way to develop an extraction procedure for the plants to be sure, okay, when the radioactive substance is in the plant, we will recover it when we do this or that procedure. This is done already, and now everybody, as I said already, is waiting for the biochar substrate, which is more or less on the way. That is where we are now in the project. I come slowly to an end now. We are more or less right in the middle. Yeah, we have a little bit delay because of this unexpected findings in work package 2. We thought because we have this yeah, problem with the composting process, which was really unexpected but very interesting, and also what we know already now, an improvement. Yeah, so it was bad and good at the same time, I must say. And we are still confident that we stay in the project schedule, though this work package 4, 5, and 6 are a bit delayed, but we think it will work. And of course, we have challenges in the project, as every project nowadays. The project is running already since 1.5 years, and of course, we were not able to make just one face-to-face meeting. We know us all via video conferences here, which we do frequently, but I would also like to meet my partners at some point in reality. And yeah, we will hope this year it will be possible. Okay, that was it with the project overview. If there are any questions, I don't know how that is organized. If you put the questions now or later, Melissa, you might help me.
Thank you very much for this great overview. I think that if there are some questions, you can try to answer now because you have to leave I think in half an hour. So please type any questions in the chat or in the Q&A panel. I will leave just in a few minutes.
It could also be if there are later questions. I don't know. Probably, I think you will share the slides. Probably, yeah. It's also possible always by email. You see my contact email address,
sure.
And we can also answer that later if you have later question. Could be.
I can see any questions. So if you have other questions, you can write an email. We will share these slides, so you will find the contact.
I think it will be more questions later when it goes into detail in the other talks, because sure, this was an overview. I hope you know a bit now where to sort the project, and yeah, thank you very much.
Thank you. So I
stop sharing.
Yeah, thank you. So it's now the turn of Dr. Joshua Mark from Peter Lab in Jerusalem, and he would talk us about from wastewater treatment plants to our plate, the whole picture of exposure to wastewater derived pharmaceuticals through food. Please.
Hi, good morning. Can everybody hear me?
Yeah.
Okay, great. So it's a pleasure being here. I had the pleasure of meeting Dieter years ago when I worked as the coordinator of the Center of Excellence in Agriculture and Environmental Health of the Hebrew University. And at that point, we already had some thought about a project like this. So thank you very much, Dieter, for the overview of the whole project. And it's very interesting to see. But sometimes when we start with the questions and every partner is going to give detail about its part in the working plan, maybe this is going to shed light for the people for the audience of this webinar today. So we are going to talk about from wastewater treatment plants to our plate. we're going to try to show you today the whole picture of exposure to wastewater-derived pharmaceuticals through food. I'm here today as the chief scientist of Fitor Lab in Jerusalem. However, I have been involved with different projects like this at the Hebrew University, who is also a partner, subcontractor for this project. So the problem definition has already been explained here by Dieter. And what happens is that we have between the urban environment and the agro environment, there is a communication which goes with biosolids and irrigation water, and that's where we have these residues of pharmaceuticals coming from one place to other. The background here, and this is, I think, one picture is worth 1,000 words, and this is one of the projects that we have published when I was still at the Hebrew University, working with Professor Benny Hefetz, who is now the Dean of the Faculty of Agriculture of the Hebrew University. And in this picture, actually have the whole picture. We have the whole picture. The paper that we published at Environmental Health and Environmental Science and Technology was irrigation of root vegetables with treated wastewater. And we were evaluating the uptake of pharmaceuticals and the associated health risks. We start here in the upper part of the picture with the food chain. The food chain goes to, after being metabolized in the body, goes to the sewage and goes to the treatment plant, which we have the residues of pharmaceuticals. Here you can see some molecules of carbamazepine in this case. And after being treated at the treated wastewater in the plant, it goes to the field where we have the uptake of this treated wastewater and the residues of these pharmaceuticals through the plant, just like in the previous presentation, we were told that the plant uptakes the labeled compounds. The same happens with the residues of these pharmaceuticals, and then it goes to consumption. So that was the first time that we knew that these contaminations happens in plants. And then this is another project that I was also part of. It was the human exposure to wastewater-derived pharmaceuticals in fresh produce. We had here a controlled trial focusing on carbamazepine. What happens in this proof of concept study that was also published at Environmental Science and Technology is that we see that the reclaimed wastewater used for irrigation comes to all kinds of crops in the field, and these crops are taken by the population. And our goal in this study was to see that if we could detect the carbamazepine in this case, which was in this controlled trial focusing on carbamazepine, was to see if we would find metabolites or carbamazepine itself in the urine of these volunteers who were fed with these crops during 10 days. The concept, the details you can see actually in this paper that will be shared, these slides, and you can look for it and later on. So we had two groups of people here, and they were both receiving fruits and vegetables that were offered by us. We were controlling. They were not fed only on these crops. However, it was added to their diet during the time of the experiment, of the clinical trial. And what happened is that we gave part of the group crops that were irrigated with reclaimed wastewater, and part of the group received crops that were irrigated with fresh water, not being recycled whatsoever. And after a week, we changed the groups, and we gave a washout period, and we changed the groups. And it's amazing to see at that point that we could detect after 3 days, of taking the crops in their diet, we could already detect the carbamazepine in their urine. And that was the point that we said, okay, we have a problem, which it's not a big problem at this point. We have not much knowledge about what are the health effects on the population after a long time period. But we know that the residues of these pharmaceuticals come to the produce, to the agriculture produce, and also come to the population. But do not worry, if you want to stop taking your carbamazepine and start taking vegetables instead, you need a ton or a whole truck full of vegetables every day to get to 500 milligrams. So it's not something for us to get worried, but it's some kind of a red flag stating we should do something to prevent these carbamazepine or lamotrigine or other pharmaceuticals to get to the population in the end. So the importance of the study is that we have important data regarding the level of human exposure to carbamazepine by consumption of commercially available produce in Israel. We have a variance in exposure levels between subjects of different groups, and we could estimate the potential risk to vulnerable populations. There is a lot of work that has been done. For example, part of the if we get people who eat roots, we have carrots and we have potatoes, sweet potatoes, for example. They are different crops and they absorb in different ways. So the variance in exposure levels in this subject it's what's going to give us the risk for these different populations, and that data that is going to be generated could guide policy decision, like what's the amount of water that can be used for for irrigating the crops. But not only that, before we get to that point, we should also see how we can make this a remediation to this problem. So the mission and the project goal has already been said by, and the project partners has already been said by the first presenter. And in this slide, I'm going to show you in the working package 1, 2, and the subsequent working packages where FITOR lab is going to be part of this project in the whole picture. So in the working package 1 and 2, FITOR has done its part by sending the samples to Germany, but the main and the most important part is going to happen here in working package 5 and 6, that we are going to have the outdoor lysimeter and the field trial in Israel. And we are going to see if the results are the same that we have in the greenhouse trial in Germany. So the combination of the results is what brings this project to be so special. And at this point, I would just like to talk a little bit about the pharmaceuticals in soil solution, which is the first results that we have of the results that we have, the analysis of Fitor and the Hebrew University. So for you to have an idea, we have here analytes by group. We have a group of antimicrobial agents, antiarrhythmic agents, anticonvulsive agents, and we can see the number of these different classes of pharmaceuticals that were found to be present in soil and also in the crops. Here we have the way that we do the sample preparation, and as we are going to share this slide with you later, you can take a look at all these analytes and groups, and the limit of detection and the limit of quantitation that we have. And what is interesting for us to see here is that the different levels of organic matter in the soil does have a difference on the uptake by crops. And that's why we chose this first one, the one with less organic matter near us. That's one that we sent to Germany because the results show, we are going to see here near us, that carbamazepine and sulfapyridine, they are found much more, and they are absorbed in this kind of soils than it's in different soils with more organic matter. As we can see here, for example, in this first one near us, we have the highest amount of not only carbamazepine, but epoxide carbamazepine and dihydroxycarbamazepine. They are metabolites of this molecule that are also active from the pharmaceutical point of view. So since we saw that the treated wastewater and biosolids were uptaken by lettuce leaves in this case, we chose the near us to be the soil that's going to be studied for the whole group. So I would just like to thank you very much for your attention. To make the long story short, the participation of Fitor in this project is by the first results showing that this is a problem in agriculture, and how much we could find of each sample, like carbamazepine and lamotrigine. However, we stop here at the problem. The solution may come from Rene and Dimitri and from the people who are going to work with the remediation, which is the new kind of compost, or that we are going to hear about it very soon in the continuation of these presentations. Thank you very much for your attention. And if you have any question, I would be available to answer.
Thank you very much for your very interesting presentation. I think we have already a question.
Yes.
is the carbamazepine a metabolism byproduct, or was it a case of carbamazepine was directly consumed and also excreted without change?
Yes, I can see that,
Okay.
that question from Taylor M. Well, carbamazepine, it's a very stubborn in the environment, and the carbamazepine, as you could see in one of the last, let me try to go back. Yeah, here we can see that we could find not only carbamazepine itself, which had not undergone metabolism, but also the epoxy and the dihydroxy. Those are metabolites. And to our surprise, carbamazepine was taken by the patient, was excreted as carbamazepine, was in the water, in the treated wastewater as carbamazepine, uptaken by the plant as carbamazepine, and detected again in the plant as carbamazepine. So we could detect carbamazepine and two main metabolites. But what you see, it's still the carbamazepine that was produced by the pharmaceutical industry and went all the way through without being changed and without being metabolized. I hope this answers that question.
Thank
Okay.
you for your answer. There are any other questions?
Yeah, so he says that carbamazepine sounds like an excellent indicator. Indeed, so that's why we chose it to be very efficient throughout the whole process of the project. It's going to be only one indicator. As we have seen, we have hundreds, if not dozens of chemical products and pharmaceutical products, but this sounds to be the most efficient indicator for us. Thank you.
Okay, it seems we have no other questions, but in any case, I think that you have left your contact in the slides, so people can use this to ask you any other questions later on. Thank you very much for your presentation. Then I leave the floor to Dr. José Julio Ortega Calvo from CSIC in Seville, and he will tell us about the bioavailability of organic chemicals in soils, potential regulatory aspects. Please, the floor is yours.
Thank you, Melissa. Good morning, everyone. Good morning from Seville, from Spain, from the south of Spain. I wish we could meet in person, but alternatively, this virtual workshop gives a chance also to interact from our office homes. So my role today here is to show you the role we have in residue project in relation to the bioavailability of the chemicals of concern. I will make an overview of why we focus in this subject and where we are coming from and where we would like to go in the process of this project. So the thing is that bioavailability science is really a very strong knowledge body of knowledge. There are thousands of publications, But in analysis that we started a few years ago, this was published in SATI in 2015, we realized that not all this science has found its way into regulation, in the environmental regulation. No? So in this exercise, we examine a bit the concepts of bioavailability of organic chemicals and how this could be used also in regulation. This was work by different group of stakeholders coming from the academia, also from the industry, and also from the regulatory institutions. For example, the ECA, the Environmental Chemicals Agency. So the thing is that this was a challenge. I mean, in the science field, we are trying to understand the world, making the concept model, different models, different definitions that well has been some part of the story when going into regulation. But in the other side, we face the need of the regulators to make very clear-cut decisions on what is risk, what is not, what is dangerous, what is not, assign responsibility. So a communication exercise was necessary, clarify and simplify when was possible, simplify. And you know that for scientists, sometimes for us, it's difficult to make this type of simplification, but well, the idea was try to, as has been said before, improve the communication of this in this subject. So later on, 5 years later, you can find, if you like to go and update, you can go to this book on different actors in the field, on the different aspects of sorption processes, toxicity, and risk assessment methods to measure bioavailability. So this is, as you know, is a strong field of research and knowledge, and our aim was trying to connect also with our problem in the residue project, with the motivation that you have already heard before by Dieter Hennecke and Yeshua Mayo. So coming a bit into this topic of how we understand bioavailability. I mean, there have been different definitions like bioaccessibility, bioavailability processes, and mainly all of them deal with this kind of processes that affect the exchange, the phase exchange between the sorbed fraction and the aqueous phase. And also is very important, this transfer of the chemical, of the released chemical, this transport into the cell membrane, and is also possible the direct uptake from the sorbed phase directly into the cell membrane, talking about many kind of biological entities that can come from an animal to a bacterium, etc. So this is a very general frame. So these are different processes that, depending on the scenario, have been defined as bioaccessibility, bioavailability, but in general terms, we can find the concept of bioavailability, also connecting the chemical activity of the compound, and using different methods to determine, and this is what I will try to explain a bit later. In terms of biodegradation and persistence, as I told you, this is where we are coming from. And we have paid strong attention to this process in terms of biodegradation processes, mainly also connected to bioremediation processes. So we found interesting connections with how to modify this phase exchange through solubilizations with surfactants, biosurfactants, DOC, dissolved organic carbon, even the relevant role of bacterial mobilization into this transfer path, really to as a mean of acting on bioavailability. And even we paid attention also to this direct transfer of the chemicals from the sorbed phase, and then later on, as you can see here, the compound can be processed biologically in terms of biodegradation by different growth promoting processes or co-metabolism, for example, etc. So this is a bit of the science we are coming from, and the idea is also go into this regulatory motivation, which is really our exact place in the residue. Then later on, we made this proposal in this ISANTI paper in 2015 that was taken in the ISO method for bioavailability. Yeah? And in this, as I said, simplified model for regulation, you can see that the different fractions of the chemical in the soil or sediment that can be from non-extractable residues. These are well-known fractions that don't really create a high risk. And then to very slow dissolving that can be measured through total extractability. So, and going into the fraction, which is really what we call the bioavailable concentration. They can include, depending on the approach, the rapidly dissolving fraction and the equilibrium concentration, the freely dissolved concentration, depending on what we consider. Both approaches are valid depending on the situation of what you want to understand. Then you can measure by desorption extraction with this ISO method, or also by passive sampling, which is also well known. Basically, you can also connect with biological tests, including biodegradation, toxicity, etc. But in this ISO method was really focused on desorption extraction with Tenax and cyclodextrins. I will go to that later. So one of the two approaches, which is passive sampling, the other is desorption extraction, basically is different as to desorption extraction because it's equilibrium based. Passive sampling is you can insert these fibers of different materials. You can into the sample or ex situ or also in situ sampling, and you can measure bioavailability as the freely dissolved concentration. Okay? It's very well established concept, and in connection to bioavailability, you can make equilibrium partitioning studies, and you can make very sound determinations of what is happening in terms of bioaccumulation. No standard method is still available. I mean, there are some attempts for standardization, but for the moment, this is the situation. So, and the second approach is desorption extraction, is the one we are using in the residue project, is based on this ISO method, the International Standardization Organization method that has published in 2020, and using Tenax, which is a solid material that can sort of sorb the compound from the aqueous phase, and you can use the bioavailable fraction as the rapid dissolving fraction after 20 hours of extraction, which is really a standard procedure. You can use also, you can determine a solid phase concentration, and theoretically you can go for chemicals with a octanol water partitioning content below higher than 3, relatively hydrophobic chemicals. You can go even to solubilities of 1 gram per liter. This was validated with these chemicals, hydrophobic chemicals, and to our knowledge, you can find better theoretical support for Tenax extraction in terms of making the kinetics of the section with Tenax, which hardly interact with the soil, and done with cyclodextrin. But with cyclodextrin, you can also determine this bioavailable fraction. So basically, just for you to get the message home, these are the two approaches: the passive sampling to the left and the desorption extraction to the right. So passive sampling, you can compare sort of an iceberg, no, which is floating in equilibrium by desorption, and then the exposed part of the iceberg is the freely dissolved concentration, but you can see that most of the chemicals are in the dissolved fraction. But again, giving you the equilibrium state of the floating, and then this is what you see, and this is what you measure, but this is really what you can estimate in theory. And the other approach is that you don't have equilibrium concentration, like this iceberg, which is being melted by the sun, and then you have a very, very small exposed part, which is just what is resulting from the dynamic exchange, and this is what you determine, the rapid dissolving fraction, which is giving you the risk taken to this iceberg that could be to the ships. So this is what you don't see, but which is giving you the risk, and this is the slowly dissolving fraction that could be in the bottom. This is more or less the theory. Still, we need more research on really what is the risk of this part that you don't see, that can also make some risk, but not so high as this one, which is the priority, and also you can see that could be also risk for that. So this is for you a bit to understand the two ways of looking at these methods. And the situation is that very quickly, I don't want to spend much more time, the two methods, the times for performance for the passive sampling, you need weeks to arrive to equilibrium, and the rapid dissolving you can use in 20 hours. You have the measurement. There is a consensus that the passive sampling can be used with sediment risk assessment, and the desorption extraction basically gives you better picture for soil, because of this desorbing and the absence of equilibrium in the soil. And the applicability can be ex situ for the desorbing fraction, the desorbing extraction. The output is in aqueous concentration for the equilibrium sampling, and then in solid phase for desorption extraction. This is the situation. I mean, we have a good standard, this ISO standard for desorption extraction, and the other thing is that for the moment is included in the regulation for passive sampling. I will show you an example. This part is now ongoing. Still, you can hardly find really situation where this has been used in the regulation for the moment, or there are some countries that can use for specific assessments. So how to use it? As I said, no, we can have also two situations. One situation, which is for the legacy pollution, where time has passed, and then the industry, the activity, or whatever has left a chemical or a site which is polluted. Then we have this retrospective risk assessment and management and how to use that. So the idea how to use bioavailability in this retrospective risk assessment and management. And this is one example. This is already in the US EPA that published this report. If you want details, you can have access to this. And basically, the idea is to use passive sampling really to make site-specific assessment of the pollution there, taking into account the organic carbon of the sediment, the pollutant already there, and the different sorption capacities that you may have according to the carbon which is in the sediment, that can be organic carbon, natural organic carbon, or also black carbon, which is higher specificity. So the KOC, the theoretical KOC can be used to estimate what you can expect in the aqueous phase, and then you can go with the passive sampling to really determine what is there in the site, specific site, with passive sampling, with this what is in equilibrium, and then according to the carbon in the sediment, you can make an estimation of the organic carbon partitioning coefficient of that specific sediment that can be different to the theoretical KOC that could be taken into calculation. So in this type of approach, what you can see is that taking into the concentration in the sediment and the real KOC in the sediment, you can propose a remediation target taking into account the real capacity of the soil to absorb the chemical. In terms of prospective risk assessment, we have also a different situation. Here, let's say, this is where we are in the residue project, new chemicals, new procedure. We want to know what is going to happen in terms of risk. So, for example, the approach we are testing in the residue. So basically, the idea is that there is nothing done in prospective risk assessment in terms of bioavailability. So in terms of regulation. So the idea was to have a proposal. We had this in this book that I showed you, is to connect this type of approaches that we propose for retrospective risk assessment to put into a prospective mode, connecting these methods with the OECD tests in terms of persistence. So our approach in the residue project is to combine these bioavailability standard tests with biodegradation standard tests, really to develop this connection between these two tests to have a regulatory motivation. The results we have gone so far are preliminary. I just will show you just a few examples what we have done so far, but you can have a fresh idea of where we are now. Dieter Hennecke has commented to you where we are in the process execution, but just to be very direct, what we have done is to test this method with C14 labeled chemicals, including pyrene and carbamazepine. Pyrene is in the chemical window already described for the ISO method, for the 10x extraction method. So we have used this ISO method with this 10x extraction and this OECD persistent assessment, really to see how these two methodologies can fit to each other and the type of adjustment we can make. And we adapted the ISO method to carbamazepine, which is at the edge of log KOW of 3, where these methods can be adapted. So what we have done is to adapt the methodology in terms of the 10x to soil ratio, so having a bit of more trapping capacity in the system to better get more results for bioavailability. And the results are shown here. To the left are the pyrene, C14 pyrene, and to the right are C14 carbamazepine. And making very short description, this is also going to be presented in the next CETA conference in Copenhagen, is that with pyrene, we could see that in terms of the inoculum, we made an inoculum in the soil to accelerate the co-metabolism of pyrene, so no mineralization. So you could see here that pyrene was was co-metabolized more or less to 50%, and this co-metabolism resulted into a higher non-bioavailable fraction, as you can see here. So the result is that co-metabolism, differently what we thought, is decreasing the risk of the chemical due to the incorporation of this metabolites into the soil, and then are desorbing very hardly. And then to the right, you can see the results that we have obtained with carbamazepine. These are very preliminary, and what you can see that after very short incubation of 7 days, what you can see is that the transformation of the carbamazepine is giving you an increase in a non-bioavailable fraction, which is recovered by oxidation. So since that the tendency is that very little mineralization, this is in line of the previous literature, the transformation also seems to go into the right direction of decreasing risk. So the method seems to be working. So we will go on with this research, and we are ready to receive also samples from the residue project, also to have these chemical measurements of bioavailability in the different soils, different conditions. So the conclusions is that we can have more realistic of assessment with these bioavailability tests. We can also jump into regulation. This science is ready for that. We have a lot of knowledge, and simplifications are possible, doing carefully for sure. We can use C3 or FRAP, freely dissolve or rapidly dissolving. We have gone into the rapidly dissolving fractions, and we are integrating that in the OCD persistent test, so we have a more regulatory strength to come and to show the evolution of risk in our residue technologies. Okay, so thank you very much.
Thank you for your great presentation. Can you see the Q&A panel? I think we have just a question for now.
I think there's something in Q&A, isn't it?
Yes, yes. Can I go
Yeah.
through that? Yes.
Yeah.
Okay, so I have found one in the chat. That's the right place. There's one question in the question and answer. A copy of the scientific paper, Regulatory in Australia, Science and Regulation is. You are a regulator in Australia. Very nice to talk to you. Sure, just write me, and I will just send an email. I will proceed. We send you relevant papers. And in the comments, there is interesting that no definition of bioavailability include absorb or toxicity to organism. You are right. You are right. Making maybe due to time restrictions, I could not make a jump into a general discussion, but maybe you recall the general scheme of the different bioavailability processes, and one important part was once the chemical cross the biological membrane, all the things that go afterwards. No, the key point here is that we are focusing on environmental risk assessment. So what is going into the water phase is our concern. Okay, and for sure, bioavailability will include all the biodiversity of mechanisms that are affecting toxicity and so on, have to be taken into account. But in terms of regulatory, the first step sort of something that we can start with could be a chemical assessment of what is going to the water phase. And this is where is placed this ISO method on really, and if you go into the definition of this ISO method, you go to the water phase, then you may have the most of the right impression of what will happen with bioavailability. But you are totally right. The bio, and believe me, I am biologist, so I really support that. The bio part is very important, but everything going through the water phase, again, this is a simplification. Because, I mean, let's say a bacterium sitting of a sediment, a grain, will take directly. This is what I showed also. But in terms of general regulation of what is giving you the right to say this is dangerous, this is not dangerous, first look at what is happening in the water phase. Okay, thank you.
Yeah, I don't think we have other questions. We can take just 5 minutes at the end of the other presentation if there's something other questions. Thank you very much.
Okay, thank you.
And now I am going to present Dr. Dimitri Drapkin and René Schaffen from the working group of geoecology of the University of Berlin, and they will talk about biochar and its use and amendment in the composting process. Please, the floor is yours.
Thanks. First, I want to start sharing my screen. No. Okay. So I hope everyone can see this and everyone can hear me.
Yes. Yes.
So yeah, hello from Berlin to every one of you, and thank you very much for having us. And thanks, Melissa, for your very kind words and introduction. Yeah, I'm René Schatten from Working Group Geoecology from Free University Berlin. And together with my colleague, Dimitri Drapkin, we will present you a talk with the title Biochar and its use as an amendment in the composting process. And yeah, we have divided our talk into two parts. First, I will give you an overview of biochar in the composting process in general and our experiences here in Berlin with co-composting of biochar and different research projects. And afterwards, Dimitri present you the approach and first results regarding the carbonization and composting of sewage sludge, which we carry out in our research residue project. Yeah, so let's start with a short introduction. It's common knowledge. Everyone knows of you that humanity is emitting too much carbon dioxide, and in 2022, this is maybe here we stand about 55 gigatons of carbon dioxide equivalents per year. And if you look at this red curve over here in this figures, it shows the business as usual scenario using conventional mitigation techniques. And this is clearly, this is so clear that we won't fit the below 2 degrees value goal. So we need no longer just to reduce the carbon dioxide emissions, we must actively remove carbon dioxide out of the atmosphere. So we need NETs, or negative emission technologies. And this slide shows some of these technologies. Since 2018, the IPCC lists in its special report 6 NETs, and they all have sufficient potential for carbon sequestration under current of foreseeable economic conditions. And one of these NETs is biochar, and this is really great, I think. So there are much opportunities, and this NET biochar stands not alone. Of course, it's connected to with other NETs, as you see here in this figure, and mainly by soil improvement or by the utilization of residual biomass, which comes into the production of biochar. Here's the link. You can have a look on it. There's a white paper published by biochar industry, and you can, it's free. You can look at it. So, but what is biochar? Just a short slide. It's a product of carbonization of biomass or other residues. I will come to that later. During the composting, during the carbonization of biomass, sorry. In this case, it's lignin-containing plant residues, but for sure there are much more other interesting input materials. So you can carbonize these materials in pyrolysis plants, like shown in this figure here. The left one is our pyrolysis plant in the Botanic Garden Berlin, and here's another enterprise producing such carbonization plants. There are a lot of more in the last 5 years, I think. So the market is growing. Also industrial facilities and the communities who are working with biochar. So biochar is very, very porous. It's carbon rich and very stable because it depends on the highest treatment temperature and the heating period, and this is very important to do this process very well, because the feedstock and the carbonization process or the pyrolysis process, this one is the key for a good biochar. And biochar, as you can see here, is a close-up SEM, is, because of its properties, excellent carbon storage and excellent nutrient storage, and it's very useful for water retention. So in the Amazonian region, it's found that the pyrogenic carbon is a key component of the high fertility terra pretas. And then it comes up that in the last 10 or 50 years, it came back into the focus, and and the research activities explode. So and we are with them, and we will continue. So but our conclusion is, because there are a lot of research was done in the last years, one of the conclusions is that biochar is not so good to use it pure, because it's a little bit nutrient poor, and it's for example dry and very stable. But if you combine it, if you mix it with green waste or other biomass, with nutrient rich and wet biomass, then you can create a really high fertility substrate or a biochar compost. And this is the most promising approach, the composting approach, and it's published in various publications during the last years. The biochar compost can then apply to soils and turning these soils into a larger carbon sink. For sustainable production of biochar, the European biochar certificate, the EBC, was developed, and this is constantly adopted to new knowledge. So there is a positive list of permitted biomass you can use for the production of biochar, but this list will continually updated, and actually currently it contains mostly of wood or woody materials or plant residues coming from agriculture, forestry, landscape management. But it's interesting that also non-plant biomasses, for example, sewage sludge or livestock manure, will be included in the EBC feedstock list, maybe in the mid of this year. It depends a little bit on the review of publications, and then this list will be updated. This is very interesting for our project, of course. Yeah, it will open up for new feedstock. And as you can see here, as I said, what we know is that woody materials, if they are processed like drying, shopping, sieving, the result is a very high quality biochar. And this meets the EBC quality criteria. We looked at, in a few projects, we looked at foliage. We made pellets out of this and carbonized it. And yeah, it is possible to reach the quality criteria, but it's not listed at this point. It's the same for animal manures. Yeah, we pellet this and we created biochar out of it, and it's not listed, but it is in process. And it's the same with sewage sludge. The challenge is always to reduce the water, the mineral components, and yeah, and maybe also the heavy metal content or the pharmaceuticals in the feedstock and the biomass. But we are on that in our project, and we look in the future. So what you see here is a short overview of the composting process. You can see it. As I said, here are the residuals, and you can add the biochar with 5 or up to 20% by volume. This is our experience. Maybe there someone of you has had other experiences with this. We mix it directly in at the beginning. This is the composting step, and then the composting took 8 to 12 weeks. There are some important things to consider. At first, while setting up these heaps, you have to adjust the CN ratio. It might be approximately 30:1. This is a good composting CN ratio. Then you have to, to shred the biomass residuals, so they are much more available for the microorganisms. The moisture must be optimal, so something about 40 to 60%. You can adjust it during the process, and the mixing is very, very important. Not only by a wheel loader, much more better is this compost turner. I think I know this is the small volume heaps. I know there are composting sites with much more bigger heaps, so it might be a little bit difficult to turn these heaps, but it's important to get the oxygen in the heaps, to continue the composting process. What is really important is to check regularly the smell, the moisture, temperature, the CO2 pore concentration, and if necessary, other greenhouse gases. I know it's more important for research project than for daily stuff, but I think if you want to have a good compost, then you have to check these parameters. The next slides, I will show you some of these composting process parameters. These parameters were collected in several compost trials in the context of various research and development projects we carried out here in Berlin in cooperation with the Botanic Garden Berlin and the Zoo Berlin. They do have a lot of waste materials or different feedstock, and we wanted to close material cycles. We want to make it more sustainable to get the nutrients back into the circle, and not only burn the material. And this is what is important about this. Yeah, and the individual names of these compost variants is not that important for you to remember. It's more important that I show you the effects of biochar. This means the variants, PK means biochar. This is, sorry, it's a German caption, but I think you can made it if you knew if you know that PK is BC for biochar. The 0 stands for compost heaps without biochar, and 5 is 5%, 10%, and so on. And so the squares and the dots without a filling are always without biochar, and those are filled are the variants with biochar. So you can see there are two variants or two compost trials. This one is in black, and this one is in blue. The axes are a little bit shifted. Sorry for that. It's unusual, but so you can divide, or you can divide between those. Otherwise, they won't be fit together. What you can clearly see is that the addition of biochar or the compost heaps with biochar reaches higher temperatures and enter the heating rotting phase earlier. So this means those variants of biochar running the composting process faster. They are better hygienized, and this is really good for the whole composting process. And this was very surprising at the beginning for us 10 years ago, or 8 years ago, I think. And this is one effect of biochar, that the composting process runs better, hygienization runs better, the temperatures are higher. And the variants variants with biochar, this is shown on this side, have a higher water content during this higher water holding capacity of biochar. You can see here the variants that are filled, or the plots that are filled, the dots and the squares are the variants with biochar, and you can see they are always on this point higher than this one without biochar in general. Over here you can see the carbon dioxide pore concentration. The same after a few days. It's always the case that this variance with biochar show lower CO2 concentrations in the pore concentration, and this is the microbial activity, and it's much. This is really good for the emission of carbon dioxide. This is lower in the variance with biochar, and you can see is, of course, not surprisingly, is the total C content or total carbon content in the compost heaps with biochar. Of course, the carbon content is higher than in the heaps without biochar. So this figures, I know it's fully packed, but they show another greenhouse gas, the methane, the pore concentration on this side, and here the release during the turning event, or the turning process per each heap. We measure it before and after the turning, and it's the same after a few days. After a few days, 7 or 8, the compost heaps with biochar show a significant lower methane pore concentration than the variants without biochar. And this is exactly here the case. Here it's not different, but after a few days you can see the effect of biochar, the absorption of CO2 at the surface, and you can see lower emissions of methane. It's the same for hydrogen sulfide. It's important for the working safety. And you can see the same result after a few days. The variants with biochar show lower H2S pore concentrations. Also, it's the same after the turning events. So this one shows you another parameter, the total mass of organic matter per each compost heap, and it shows you at the day 1 at the beginning and after 191 days of composting. This bar over here is the variant without biochar, and here are 2 biochars added. It's not so important what's the difference between them. Important is that they are starting at the same point with the fraction of the organic matter biomass, and the biochar content is on top of this, and this belongs stable during this time. But you can clearly see the degradation of organic matter was much more higher without biochar than with biochar. You can see it in this this difference over here. And this means after the whole composting process, there remains more ready compost with the adding of biochar. And this is quite good, because if we want to have more material, then just add biochar and stabilizing the degradation process. Of course, we characterize our produced biochars physico-chemically. Here you can see this in the table. There are a few biochar listed. This one is always without BC0, and here are 5 and 10% biochar added. And these are much more biochar compost from an older project, our first biochar project in Botanic Garden. And here's the mean value and the standard deviation. I think there are 10 or 14 different biochar composts. Is the range of it, and the mean is, I think it's mean. And you can see here the value range of our Federal Compost Quality Association here in Germany, and the quality criteria for substrate compost. And it's important to hold these range to stay in this criteria for substrate compost. Then you can use this in a botanic garden or elsewhere, and you can see, of course, the pH rises up with adding biochar. It's in the range of these quality association, and it's the same for the salt content, organic substance, and the nutrients. Also, the plant available nutrients, they are okay, they are in the range. So this compost produced with biochar can comply these value ranges, and they can be classified according to this quality criteria. It's in Germany, it's substrate compost type 1. You can use it. And another good result is that we look for the leaching behavior of these produced biochar composts. And I think it's also a good result. The adding of biochar leads to a reduction of nutrient release. Here you can see it in comparison to the substrate without biochar. It's this line, and everything on this side is the reduction of the release in percent. So nitrate is released up to 50%. The nitrate release is reduced up to 50%, phosphorus and the potassium release as well. And it's increased with aging this biochar substrates. So we carried out batch and column tests, and it's the same in both methods. And we tested it for every produced biochar. And this is, of course, quite interesting for the fertilizer amount and for the protection of groundwater and drinking water if the nitrate release is reduced. So I will sum up our experiences here in the Botanic Garden or at Free University. Yeah, over the last years, we do have higher temperature during the composting process. It's a better organization when you add biochar. It's the moisture is reduced. This means the free water is reduced. In the final compost, of course, there is a higher water content because of the water holding capacity of biochar. But during the process, yeah, the water or the moisture is much more stable, and this is quite good for the composting process, of course, for the microorganism activity. And yeah, another result is that the smell is reduced, and the emission of greenhouse gases, as I show. There's a reducing carbon decomposition. The structure of the ready produced biochar compost are much more better. It's more crumble, and the nutrient leaching is significantly reduced. So I think this, yeah, it makes sense to add biochar to the composting process. We saw no negative thing, and this was quite good. And until now, till this day, the Botanic Garden in Berlin uses biochar for its composting process. Here, yeah, I will make this quick because Dimitri wanted to tell something about residue, I think. So these are our 4 biochar projects. Yeah, some we carried out and one ongoing. I will make this really quick. Maybe you can have a look at this later. This session is filmed, and so you can have this later. We made some new composting process in the Botanic Garden. This is a good goal in the Terra Boga project. Then we added biochar compost on military conversion areas and low yield sites to improve soil quality. We went to the zoo in Berlin and transferred the knowledge of Terra Boga to the zoo. Then there we had another input materials like animal manure or the foliage. It was very interesting for us, but it worked. We produced nutrient-rich biochar compost. And our new project, this is still ongoing, is a project here in Berlin where we look if the soils in Berlin are carbon sinks of the future. And this is quite interesting because these are former sewage fields, and we want to look the suitability of the soils as sea sinks. And we are also testing carbon rich biochar substance in street tree plantings. And this is the ongoing. Yeah, we are looking forward. But what's new in residue? I presented all our experiences, but there are still so much more. But I have to finish now. What's new in residue? It's a new input material. It's sewage sludge, and the former speaker said it's very challenging. But now I want to hand over to Dimitri, and he will tell you more results from residue.
Okay, thank you, Rene. It was really a very hard task to press all the results from a bit more than 10 years of research in our group here in Berlin into a short presentation. So I just now need to summarize that we, in our group, we had promising results, mainly dealing with green waste compost. But in residue, of course, we are dealing with a totally different material, and the motivation was, to to transfer this promising results concerning biochar amendment into composting process, dealing with composting of sewage sludge, and in the next picture, we see mainly our approach. So it shows it very nice. We are dealing with sewage sludge as a composting matrix, and we use actually agricultural residues for biochar production, and we use sewage sludge itself for biochar production here. As Rene said, actually we mainly used wooden biochar for wood as an input material for biochar production, and he also mentions that other materials are mainly still pretty difficult for this purpose. You see here in the picture, for example, food wastes or animal manure, which needs some special pre-treatment to really get good results during sterilization. Generally, we are very grateful that we could work together with a compost ore facility in Israel, where we actually could establish our trials with biochar amendment, and facility is actually the biggest one dealing with sewage sludge treatment, and they already have a composting process there going on the standard basis. And we, first of all, we thought about input materials for biochar production that we could take. Wooden biochar is obviously the most used input material, but it's in this case, in the residue framework, we pretty well aware that wood is a limiting material in Israel and also in a number of Mediterranean countries. So we looked for another opportunities, and of course, the best solution would be also that the material is also you have it in the same place where you have sludge, so it's you have also look at the economical aspects of the whole thing. We were thinking about using sewage sludge like in a two-way recycling process, where we get biochar from it as well as composting matrix, and we realize our co-composting idea with these two input materials. We actually at the first we got the composted sewage sludge from a compost ore facility and sterilized it in a preliminary tests and at different temperatures. And afterwards we also saw that the compost ore also produces biochar from its composted sewage sludge on its own in a bit different manner, because they mix additional wooden chips compounds to that input material. So we actually, as you see, we actually already have these two to input things realize that were already existent at the compost ore facility, the composting of sewage sludge itself, and producing biochar from it, which they actually use in the greenhouse experiments as a standalone fertilizer. And the new thing that we introduced now in the residue framework, in the framework of our project, was this co-composting approach with these two materials. And in the next page, you see, first of all, the results of our preliminary test with sterilized composted sewage sludge. And we sterilized it at 3 temperatures. And here you see it as in the form of Van Crevelen diagram, which shows the ratios from oxygen to carbon and hydrogen to carbon on 2 axes. And you see here the stability zone shown, where in which if materials lie in this zone, then the biochars can be seen as stable biochars, which is a quality showing quality of this biochars. And if they do not lie in this zone, then it's not so good for the characteristics of that material. And you see that we can compare it to this green dot is a wooden biochar, summarizing results from different wooden biochars from our trials in Berlin, in Botanic Garden. And you see also this standard deviation in 2 axes. And but because of course also these wooden biochars are very different materials, but anyway, this is a most stable biochar type that is produced from wooden residues. And we also in the carbo tip project, which Rene mentioned, we also had biochar produced from foliage. You see it here with in the blue dots and at different temperatures. So it's obviously not so stable as wooden biochar, but still a very good material. And also our sludge biochars, they lie in this zone, and they comparable with for example, with some foliage biochar, and it's actually a very good result, showing that this biochars produced from composted sludge are really stable and good biochars that can be used in the co-composting. In the next page, you see we analyzed these biochars that we produced concerning, for example, plant available nutrients. You see that they are much less than the original material, original sewage sludge, but still have, at least concerning phosphor and potassium, they still have a pretty high levels of this plant available elements. And this shows actually why this material can also be used as a standalone fertilizer, which is seen here on the picture in the right from this greenhouse trial at Compost Ore, where they use it as a standalone fertilizer. So this picture just to give you an idea of how this material looks like. It's pretty different from what we had with, for example, with wooden biochar, with actually a styro material not having or very less plant available nutrients there, almost nothing there. So this material can be used as a standalone fertilizer, but of course you also have through permeabilization, you have a big reduction in the plant available elements there. Okay, so in the next here we see our first trial where we first tried to compost sewage sludge based on information that we got from Israel here in Germany, but that showed us that yeah, sewage sludge is a pretty difficult material for composting, as we had almost main experience in composting of green waste before. It was very different from it, because green waste brings higher temperatures in the composting process, and if you take sewage sludge, you really need very big composting heaps to get the high temperature that is needed during the composting process. And as we operated with smaller amounts, we couldn't get this high temperatures, and actually this first trial didn't function. And also the first trial that we tried in Israel together with compost all did not function, because also there was a problem that we didn't get needed composting heaps amounts this time, because of biochar as a limiting factor, that there just was not enough biochar that could be produced for such bigger composting heaps. So this was the issue why another trial setup was installed there with forced ventilation, which in the end brought us at needed temperatures, and that we could get needed compost from that approach. Forced ventilation allowed us to get the composting process on also with a smaller composting heaps. The main idea here is that the air is pushed into these composting boxes, which promotes the microbial activity that is crucial for the composting process. And here you see this setup at compost. Oh, very nice. It looks great. And we established there our 4 variants that we wanted to study. This was the control without biochar, and 3 variants with biochar application with 10, 20, and 30 volume% of biochar in them. So this new trial, we actually, these results are still very fresh that we got from this trial. So these results, these are mainly optical and sensoric results, but they already show some interesting aspects. At the top here, you see in the first picture at the top, you see the control variant, and you see this white mold built on it. And on the bottom picture, on the bottom, you see the variant with 30% biochar. So what you obviously can see is that biochar amendment effectively prevented mold, which is probably due to reducing moisture, which we saw previously in our presentation. It significantly reduced smell. What is these 2 things, they show up to probably a better hygienization with biochar amendment. What is very important is that it significantly made the compost structure significantly more crumble. So we get better compost structure through biochar amendment. So these results, first of all, are very promising. So we also think that a new trial on a larger scale can be realized at compost ore, because of also because of the fact that more biochar will be available for the trial with the new sterilization facility. And so there are some 2 aspects of that. First of all, this material will be provided to the project partners for further investigations, for further studies in our joint residue project. We ourselves still want to characterize these materials on the physical-chemical characteristics, and also on we want to make leaching tests with them concerning mobility of nutrients and contaminants, heavy metals and organic contaminants. And of course, these materials also will be used through our partners in the study of the fate of pharmaceuticals. We will look how these materials can alter the fate of the pharmaceuticals in the trials with irrigation water. So as Dieter mentioned at the beginning, the project has to be fruitful, to give practical results. We have to show what we reached, and I think from that point of view, the first thing is our further cooperation with Compost Ore, where we can directly see if these smaller trials can be transferred into practice in some way. And we also want to present these first results in the CETEC conference in Copenhagen, and also in the framework of our summer school of Hebrew University, where residue project will have a section, and also in the specific biochar conference in Darmstadt in Germany in November. And here are our sources that we used, and that's all from our side for now. Thank you.
Thank you very much, Dimitri and me. I think we have some questions both in the chat and the Q&A panel.
Yeah. First, sorry, we talked a lot too much. I think we are running out of time. Sorry for this.
If you can answer just two questions, and then we will move to the last presentation.
Yeah, I saw the questions in the chat, and it's totally right about the carbonization process. Yeah, it's totally right as it's said here. It's without oxygen, of course, under oxygen deficiency, and then you got the biochar, and it's not burning. It's the carbonization, the pyrolysis process. It's mentioned right in the chat. It's totally right. Maybe I explained it not good enough. So thanks for the explanation here. There's another question about the CN ratio. Yeah, this is true. Biochar, yeah, of course, there has a high carbon content, and you can add freshly cutted green waste or garden waste. This is very very nitrogen rich and wet. Or we used elephant manures out of a zoo, but okay, of course it's not available everywhere. So manure from animals are also nitrogen rich. And then you can adjust the CN ratio. Is there another question? Not sure. In the Q&A is one. What is the impact of adding biochar with the bioavailability on sewage sludge?
Okay, that is we want to see in this project but this is something that will be studied through our project partners mainly.
Yeah, maybe Jose Julio and Dieter.
Yes, we are looking in the leaching on the nutrients and contaminants, especially heavy metals, but pharmaceuticals is something that will be seen by Jose Julio and of course by Dieter.
Yeah. Okay.
Okay.
And there
So
are no. I think.
no
I
further
think. I think.
questions.
Okay.
Okay, perfect. So thank you very much. And I think we can move forward to the last presentation from Dr. Federica Brogioli from D&D Biotech, which is an innovative Italian company which aims to develop bio-based technologies and proposes the use of biotechnological remediation strategies. So I leave the floor to Federica.
Yeah. Hi. First of all, can you see my presentation?
Yes, we can see it, but I can't hear you well.
Do you hear me better like this?
No, I don't know if just my problem or.
Can anybody else?
Okay, okay. Maybe now I can hear you well.
Oh, okay, okay, good. Yeah, so you see my presentation full screen, right? Okay. So yeah, hi, I'm Federica Brogioli from Biotechnologies from D&D Biotech. And thank you, Melissa and Italbiotec for inviting me and D&D to talk in this workshop. I will try to be brief since we are a bit behind schedule. So today I will talk mostly about bioremediation of contaminated soils and about molecular biology techniques that can be used to study the microbial community present in the soil and responsible for the degradation processes. About D&D Biotech shortly, is a consultancy company based in Pisa and is specialized in biotechnology and engineering of bioremediation processes. And in this presentation, I will describe how metagenomics and process engineering can be integrated together in a successful bioremediation project. Especially explaining the use of RoboNova, which is a machine developed by D&D Biotech to perform biodegradation optimization studies at the pilot scale. So biological processes for remediation of contaminated environmental matrices are based on the ability of microorganisms such as fungi and bacteria to degrade partially or completely many types of organic contaminants, for example, petroleum hydrocarbons, fuel additives, pesticides, or chlorinated solvents, to name a few. And bioremediation can be of two types, as you can see in the screen. In situ, which means at the spot where the contamination is present, or ex situ, which means that the contaminated matrix is moved to a different location and then it's treated. In situ bioremediation can be intrinsic, which means that it happens naturally and it's hosted. And this is based on the microbial community, the type of contamination present, and the characteristics of the soil. Both in situ and ex situ techniques can also be engineered. And there are different types of in situ and ex situ applications, but they can both benefit, especially from biosimulation and bioaugmentation. Biosimulation is the addition of nutrients and oxygen to the soil to be treated, while bioaugmentation is the addition to the contaminated soil of an active microbial culture that is known to be able to degrade the compounds of interest. Bioaugmentation can be done using bacterial or fungi cultures, which are already.
Sorry, Federica, if I interrupt you, but we have some people that have problem to hear you well. I don't know why I'm still having problem with hearing you well.
Okay. Yeah, I'm not sure why precisely.
No, it's something like it's alternating from up and down.
Can you hear me better now?
Yeah, now yes.
Okay.
Okay.
Should I say something again, or should I go back?
No, okay. Can you continue, please?
Okay, okay. So yeah, so I was saying that bioaugmentation can be done using bacteria or fungi that are already present in the matrix, so they are called autochthonous, or with commercially available active cultures, so allochthonous. The high interest in the possibility of taking advantage of these natural processes in the remediation of contaminated sites is due to the potentially greater sustainability, both in terms of environmental impact and costs of the biological techniques compared to the standard physiochemical ones. However, the current best practices are often insufficient to achieve the objectives of decontamination within a certain time frame. And the reason for this is that biological remediation strategies are very site specific, and their effectiveness is dependent on a lot of factors, such as chemical and physical properties of the soil and of the contaminants present, of the mechanisms and conditions that control the growth of the microorganisms, and also the local climate, as in temperature, since most of the techniques are performed in the open air. So many variables make it impossible to create a full similarity model that can include all these parameters, and also impossible to replicate the same processes and methods on all the different kinds of remediation scenarios. So pilot testing on a significant scale remains the only real support for the design of a really site-specific bioremediation intervention. So this is why D&D created RoboNova, which you can see in the picture. And RoboNova can be considered the on-site model of an ex situ bioremediation technology, such as the biopilot. The technology at the basis of RoboNova is based on the optimization and exploitation of aerobic processes performed by microorganisms native to the matrix under treatment, which are, by definition, able to consume the organic contaminants present in the soil together with oxygen to sustain their growth. This is true for a large number of compounds that have been studied so far and that are also researched on a daily basis. Of course, without intervention from outside, from external modifications, the degradation is generally quite slow, since key parameters such as nutrients, oxygen, and water content are not optimized naturally. The pilot, or RoboNova, allows to model the intervention to be performed on site, working on the specific methods to be treated. The machine, as you can see, is placed in a transportable closed container, allowing a relative control of the temperature and shielding from sunlight, preservation of the soil from rainwater, regulation of humidity inside the soil piles, and reduction of leachate production caused by rainfall, erosion caused by atmospheric agents, and spread of dust odors. The container is also equipped with an air filter, a water treatment unit, and a fermenter, which can be used to grow bacterial or fungal cultures to then be added to the treated soil. Generally, a bioremediation optimization study realized using RoboNova involves a number of phases. First phase is soil pretreatment, which consists of a first study of the soil type to delineate its characteristics and properties. Then next phase is addition of a suitable working agent to increase the concentration of bacteria and improve their distribution in the soil. A working agent can be fine wood or green compost, or also an industrial waste, let's say, which is mushrooms growth substrate from the food industry. And finally, the last part of the first phase is the optimization of the nitrogen phosphorus potassium ratio, achieved by adding nutrient compounds as well as pH stabilizers. As nutrients, usually urea is used together with superphosphate compounds, which consist of both calcium sulfates and calcium phosphates. The next phase, so phase 2, is biostimulation to help improve the natural degradation abilities of the microbial population. This phase involves aeration of the soil piles by tilling, which is performed in Robonova mechanically and automated by the soil burner that you saw in the middle of the picture. Then next step is to keep the ratio between the contaminants and the nutrients concentration, so the CN ratio, stable, and then keeping the optimal water content and by irrigation and the right temperature. Irrigation in Robonova is performed by water spraying on top of the piles, and the water that is not retained by the soil is collected in a tank under the soil pile, and it's either recirculated in the system if needed or treated and disposed of. When all these parameters are optimized, it's possible to proceed to the last phase of the optimization study, which is bioaugmentation. And this is achieved by adding to the soil pile hyperactive culture of bacteria or fungi or both that was previously prepared inside Robonova. So to prepare the culture, first you isolate the microbial population from the soil sample, and then you grow this population in the fermenter, which is in Robonova, and then you add the culture to the soil. So the use of the Robonova pilot plant to perform this optimization study allows to obtain uncertain information and parameters that are needed to design a full-scale bioremediation plan. For example, it's possible to define which machinery and instruments are needed to perform the treatment, what chemical additives need to be mixed with the soil, and in which concentrations and time frames and frequency. Then also to delineate the degradation kinetics of the compound of interest that needs to be degraded and the expected time frames to obtain the desired degradation goals. Also, it's possible to estimate energy consumption, environmental impact, and production of waste and emissions. And finally, a monitoring plan to delineate a monitoring plan, which techniques are required to follow the degradation progress. On this note, I want to introduce briefly the second part of this presentation, which is next to the engineering of the bioremediation strategy, the molecular techniques that can be used to study and monitor the microbial population responsible for the degradation processes. The Robonova pilot plant can be used to implement treatment of cycles, which are based on the results of the microbial characterization. The characterization involves sampling the treated soil and performing genetic, bioinformatic, and ecotoxicological analysis of the contaminated matrix. As you can see from the scheme, Soil samples are taken from the treatment site at different time points throughout the remediation and tested with metagenomic analysis. The analysis is based on the sequencing of the DNA isolated from the soil sample by means of next generation sequencing techniques. So it's a broad screening of the whole biodiversity present in the contaminated soil. This analysis allows to identify the bacterial and fungal species present in the sample and to characterize them. And then with bioinformatics tools, it's possible to investigate the degradation abilities of the species present in the sample and to monitor their evolution in terms of relative abundance within the sample along the course of the degradation process. Based on the results of the DNA analysis and of the chemical analysis as well of the contaminants, if needed, it's possible to modify the treatment strategy applied to the soil in the RoboNova pilots to improve the performance of the microbiome community and thus of the degradation. Add more nutrients or applying stronger aeration or higher irrigation rates. And a new cycle, a new treatment test can be programmed and performed within RoboNova. To conclude, I want to mention that the techniques that I described in this short presentation were related to bioremediation of contaminated soils, but can be applied also to other type of soils, matrices, and with some adaptations also to water. To be able to remove biologically compounds that can pose a risk to the health of an environment or animals or people that live in it. And that the molecular techniques that are used to follow the biodegradation processes or metagenomics can also be used to detect pathogenic species in water or soil sediments. So these techniques can be applied to applications such as wastewater and sludge or sediments intended for reuse for agricultural purposes, which is in fact the topic of today's workshop. And with this, yeah, it's all, and thank you for your attention. And I hope you could hear me.
Thank you very much, Federica, for your very interesting presentation. I don't know if there are any questions.
Checking the chat and the question.
Yeah, it's sorry, it's possible to receive the certificate of participation. Of course, you can write to us. I type in the chat our email. If you write to us at this contact, we will provide you with the certificate of participation, and the slides of the speaker will be published on the Residue website. I type it also in a chat. I don't know if we have some questions for our speakers. We can take just 1 minute to. I don't think we have other questions. In any case, if any question would arise, you can write to us at the email that I tapped in the chat, and we will forward them to our speakers. Okay, so thank you very much to all of our speakers for your participation in this workshop. It was very, very interesting. I hope that it was so also for our participants. Thank you very much. Have a good day. Bye-bye.
Thank you, and thanks for you and Cecilia and your team for organizing this very cool workshop. Yeah, it was a great workshop, I think.
Thank you.
Thank you. Bye-bye.
Bye-bye. Thank you, Federica.
Bye-bye.